So I've been trying to figure out what makes the human brain so amazing, and there's abilities that you have when you're sitting there and thinking about what you're going to do on the weekend, or what you have done, or what you think of somebody else, and these abilities to have this awareness and this and your imagination, all these kind of abilities are things that are very specific to our brains. There are other animals that don't have these abilities. You don't see ants walking along in a line and then one steps out and goes, I don't feel like doing this today, I'm going to go do something else. There's instinct in those animals, but for us, we have these amazing abilities. So it's trying to figure out, well, what makes this so amazing? Our brains have almost or more than doubled in size in the last 3 million years in evolution. That is an incredibly short space of time in the whole time frame of evolution for our brains to double in size. That is quite incredible, and it hasn't doubled in size just everywhere. There's only parts of our brain that have expanded tremendously, and these are the parts of our brain that actually give us all those amazing abilities. So it's really trying to decipher how this actually works and what happens. But obviously there are issues. So we go through our life sometimes, and all right, here we go. And our brain is fragile at different times and in different places and in different conditions. So you can see even in childhood, we have diseases such as epilepsy, childhood epilepsy that can afflict the young brain. And then we also have, through adolescence, time when we're supposed to be the strongest and everything is going really well for us, and we don't normally have diseases associated with us. Then we still get conditions such as schizophrenia. And then clearly when we get older, we do have other issues sometimes, such as neurodegeneration. The brain and its evolution is amazing, but things like how long we live. We now live, double the amount that we used to live just about 100 years ago, since penicillin and other great medical breakthroughs have really increased our lifespan. But our brain hasn't been able to catch up yet, I guess, in a way where it's able to protect itself from things that may happen deleteriously to our brain through aging. So it's really trying to learn how this human brain is working, and there are many aspects of the human brain that is different from other organisms. So a lot of times historically now we've been using models to learn about molecular and just the real nuts and bolts of how the brain works, through using other organisms such as worms and flies and mice. But clearly some of the aspects of our brain are not going to be found in the other organisms. So we have had some problems in trying to figure all of this out. The first one really is to have a suitable human model. So like I've said, our brain's different to other animals. So how do we look at aspects of our brain that are different? There's no way of doing this. We can't do this in mice. We can do it because we can use them in an experimental way. We can manipulate their brains a bit while they're still alive and perform experiments. Can't do that with humans. I'm pretty sure no one's going to want their brain to be fiddled with while they're still around. So that's been historically a major problem, is to try to figure out, well, how can we actually look at the human brain? There's ways of imaging the human brain and seeing where the activity is and aspects like that, but how do you look at really the nuts and bolts of how things are working? So that was the first problem. The second problem that we have is, how do we look at all of our DNA? So the DNA in all of you is made up of building blocks, and these building blocks, there's about 3.2 billion of them. And so it's been very difficult through sequencing that we've been able to do previously, where you can only look at very short stretches of your DNA at one particular time. But as I'll show you, it's obviously increased tremendously just in the last 10 years, where we can now look at broader regions, if not the whole part of the genome. So these were very important aspects of being able to really realize and understand how your human brain actually works. So this was the first really major technology, at least for me, working on the human brain, was the introduction of a way of making human stem cells. So not getting it from embryos or any of those types of historically controversial methods, but actually being able to make a human stem cell. So the way that we do this, in 2007, 2008, some papers came out that showed how to do this. And what we can do now is take a skin cell from any one of you or anybody else, take that skin cell and reprogram it into what is known as a stem cell. And a stem cell is a cell that has the ability to become any type of cell. So if you think about that, what we can do is take a human skin cell, make a human stem cell, and turn that into a human brain cell. Right? So now, if you think about that, you actually have the ability now to actually take a look at the cells of a human that is still alive. Right? So this is a major, major discovery. And the people who invented this won the Nobel Prize 2 years ago, just to show you the significance of this technology. So what we're able to do now is you're able to take stem cells or skin cells from healthy people and from patients, whichever patients you want to do. And I mean, it's also important just to take it from healthy individuals so you can see what normally happens, right? And then what we can do with these is make these stem cells. They're called iPS cells, from both healthy and controls and disease states as well. And then in this example, these are from Alzheimer's disease patients. These are from Alzheimer's disease patients down here. And there's particular parts of our brain and particular cells in our brain that are affected in Alzheimer's, and they're called cholinergic neurons. And you can make these neurons from these stem cells. And so now you can look at the differences. What's the difference between a cholinergic neuron in a control patient and a cholinergic neuron in somebody who has Alzheimer's disease? And this has now given us an amazing ability to actually now have a human model of the brain. And they've even started making little mini brains called cerebroid bodies, where you can actually show the development of a human brain from these what used to be skin cells. It's quite amazing. This is the second huge technological advance. And obviously, as Andrew had talked about earlier, having the new X10 machines in the building, which allows us now to do genome sequencing like we've never been able to do it before. The first human genome was sequenced just over 10 years ago now, and it had taken 10 years to get to that point from the start of the project. So it took them 10 years to sequence the first human genome at a cost of billions of dollars in the States. And now, today, using the X10s, we can sequence an entire human genome in one day at a cost of about $1,000. So just in 10 years, the advance of being able to look at your DNA has been incredible. $1,000 from $1 billion, and one day from 10 years. And this is only in the last 10 years. This advance now has been able to, so now we can use our human stem cells. We can see the differences. We can look at the DNA of these stem cells and see what is different. And we can do this very quickly. So the idea of medical genomics and individual personalized therapies using these technologies is a reality. And Garvin is really one of the leaders now in these technologies. So it is very, very exciting. I put this picture up because I thought it was cute, and I have little boys, so I thought that was quite nice. But what this is illustrating is that, you know, if you're driving in your car and your car breaks down and you pull off to the side of the road and you call a mechanic, hopefully he's a bit older than this one, but the mechanic will come there and go, oh, you've heard this noise, there's smoke coming out of this, but this is what's wrong with your engine. They'll fix it up, you're back on the road again, because that mechanic can take that engine, pull it apart, put it back together again. They know everything that happens in that engine, so they can fix it. For us to be able to fix the human brain, we have to understand a lot more about it than what we do right now. So it's important not only to look at diseases, but it's really important to look at what's normal. How does your brain function? Most of you, I'm guessing, your brains look perfectly fine. and so why is that? Why is your brain really good? Somebody else's brain isn't. Do we understand that yet? Not really. I mean, there's a lot, you know, using the iPS cells, using our new genome sequencing, but we now have this ability to use those technologies to really push the understanding of the human brain, of the workings of the human brain, and then when things do go wrong, and a lot of times they go wrong in parts of our brain that are newly evolved. So those parts of our brain that have evolved in us, but not in other animals, right? So it's understanding those differences. that is really going to make us understand how the human brain works, and then when it goes wrong, how to fix it. Because right now it's a bit of a guess of what's going wrong, how do we fix it. We don't know everything that's happening in it, not like this little kid. So I'm hoping this generation, including him, maybe my children, they can come through and they can use these technologies to really, really push those boundaries, really understand how this human brain is working, and then how to fix it. And I think that is really our future, and it's our generation, and it's the reality that we live in right now. So I'll leave it there, and I think Peter's up next. So thank you very much for listening.